So my name is Aaron Pomerantz. I'm the Associate Director on our Global Segment Marketing team, where I cover the non-human and applied market segments. It's been a fantastic PAG so far, an incredible number of presentations that have been leveraging the technology. So we'll go ahead and start to kick things off as we start to bring on our panel of speakers for the afternoon session to talk about the ways that nanopore sequencing is being leveraged for plant and animal and ag bio research and applications. So we'll have a great lineup, everything from rapid and scalable amplicon sequencing to pathogen surveillance applications to whole animal and plant genome sequencing all the way up to T2T. So I hope you'll appreciate as we go through the series of talks, these kind of scale up from single amplicon applications all the way to highly complex genomes, which I hope you'll appreciate leverages the unique capabilities and unique attributes of the platform. So for those of you who might be a little new, I know there's a number of absolute experts in the technology that are here with us today. And some of you have been presenting. If you caught Sergey Korin's great talk, or Mike Schatz, or many others who are involved with telomere to telomere consortia, all the way to pull-down applications, there have been an amazing number of talks here. But if you're a little bit new, we'll just go through very briefly how nanopore technology works and some of the applications and how it can be leveraged. So in brief, this is a single molecule sensing platform. It's capable of detecting a number of analytes, but thus far the company is focused on DNA and RNA sensing. And how it works effectively is using a biological protein-based nanopore. You'll notice that in blue, that's embedded in a synthetic polymer membrane. And so as such, it's acting as a gateway between two systems. So as you extract and isolate your nucleic acids, you can adapt a motor protein that you see there in purple to those molecules, and then those will then find their way to a nanopore, and they'll start to translocate a molecule all the way through. So it's kind of like a slip and slide. You'll see on your right-hand side, as a molecule is passing through that little pore, an ionic signal is being disrupted. And what's happening there is we're effectively able to turn that raw squiggle plot, or that signal, that's an electric current, back into the series of nucleic acids that were passing through that pore. So that's highly effective for a number of reasons. I hope you'll appreciate on the bottom, you'll see that offers a lot of unique capabilities. So you'll hear the words native sequencing a lot. We're not sequencing by synthesis, so we're not making copies of that molecule. It's effectively sampling and looking at a native DNA or RNA strand as it passes through that pore. So that's interesting for a number of reasons. First, you can capture very long stretches of DNA or RNA. That's where you see a lot of the technology being used today for genome assemblies, because you can span long or repetitive regions of the genome, and you're not limited by any upper read length limit. I think the record today is probably still about 400,000 base pairs, if I recall correctly, somewhere in that ballpark. 4 million, thank you, Sean, bases. Although someone came up to us at the booth and said they beat that record on coffee, so we're gonna verify that one. Another factor I hope you'll appreciate too is this system can take in short read lengths or long read lengths. It's sometimes referred to as a long read platform, but we really like to think of it as an any read length system. So if you put in amplicons, as we'll see, of about 600 base pairs, you'll sequence those for DNA barcoding, all the way up to megabase level fragments. Moreover, we can get that data very quickly. So data can be streamed as it's being base called in real time. So there's no fixed runtime on the system. It's actually a really interesting and unique attribute. You could run the system for 10 minutes, for an hour, for the full life cycle of that flow cell, depending on how much data you need. And so that's really interesting, and there are a lot of great use cases that will try to get data as fast as possible. Whether that's, say, a service provider who wants to get their data to their customers next day, or if that's rapid pathogen sequencing where you need an answer maybe in minutes or hours. Okay, and so as you may have seen over the last few days at PAG, there are a number of different attributes of that sensing system that can be leveraged, whether that's detecting SNPs or those canonical bases, detecting structural variants, and also leveraging the fact that we can detect DNA modifications like epigenetics. And so here we can detect these through all context models. My colleague Jeannie will talk a little bit more at the end about how that's being leveraged in some of the plant and animal use cases. Synthetic DNA modifications can be integrated into the system and detected. And of course, we can detect native RNA sequencing, which is the only technology that exists that can sequence a native RNA molecule, and there are a number of fascinating insights that you can gain when you don't have to convert that to cDNA first, and you can sequence the RNA molecule directly, whether that's a messenger RNA vaccine or whether that's in your actual biological samples where you want to gain additional insight or look at RNA modifications of interest. And then finally, chromatin conformation methods like POR-C, where you can get that higher level contact information to generate your complete and oftentimes T-to-T assemblies. Okay, so reflecting a little bit over the last 10 years, it was first launched as the MinION device about 10 years ago. That was our flagship product. That's when I started using it, when I was taking it into the field in the Peruvian Amazon. And since then, the community and yourselves have really leveraged it for an amazing number of use cases and applications. And so the figure on the left is actually quite outdated now. It's over 14,000 peer-reviewed scientific publications that Gordon presented on just yesterday at the JP Morgan conference was that updated figure. And we saw that just last year, over 275 plant publications and 189 animal publications. So we're really grateful that you in the community have found this a useful tool and are really here to help accelerate your biological research and applications. Okay. If you've been to any of the talks over the last several days, you've probably seen the word T-to-T thrown around. So telomere to telomere sequencing has really become a fascinating application where we're filling in all of the gaps in first the human genome and recovering over 8% that was missing, which was historically considered dark data, dark biological genome material. Oftentimes in, say, the telomeres or in centromeres or in other intractable regions with legacy technologies. But now it's becoming easier and easier to do. I think Kevin will highlight that in some cases, and it's really setting a new standard for how we look at genome information. And of course, this is being applied to a number of different plant and animal consortia and projects. And again, I think we've seen everything from okra to ruminants to several taxa in between, where the technology is really being leveraged to help provide these telomere to telomere assemblies. And as we'll hopefully showcase to you today, it can be done just on a single platform with nanopore sequencing to produce these high quality and very complete assemblies. I did say, before we start to pass it on to our speakers, I did want to mention a program that some of you may be aware of. This is called ORG.one. And this is a philanthropic endeavor that Oxford Nanopore supports the community on. How it effectively works is that users who might have access to biological material of rare or endangered species on the IUCN REN list can partner with us, and we can help to support you to sequence the genomes for conservation purposes of those organisms. And we really are doing this too, where we try to support this as an in-country activity. We do believe that the technology can really help to be more accessible, especially in environments with high biodiversity levels, but may have historically not had access to sequencing technologies that have oftentimes been inaccessible because of the capital expenditures or other factors. And so as we can see, this has been going on for a little while now. We're really excited to see what the community's done. Over 119 uploads from 105 species to date, 29 plants, 2 fungi, 88 animals, and this has rapidly been growing. So if you're interested to learn more, you can scan the QR code. Again, we're really excited to see what the community can do with these species and their conservation efforts. And actually at PAG on Friday, some of you may have been there, some of you may have presented at it, were a session that was entirely held on ORG.one. So this was really exciting to see how the community came together, and we're presenting some of their results. And this is sequencing that's being performed from Bermuda to Ecuador and other locations. Actually, just out of curiosity, is anyone participating in ORG.one here with us today? Raise of hands. I see we've got a few. So please do look around and talk to colleagues who have participated in the program. And again, you can get their experience and see how we can support the communities for enhancing more of these genomic conservation efforts. All right. So just a last few couple of updates. Wanted to touch on some of the technology and the platform updates, as well as accuracy and assembly in particular. So let's start out maybe with microbes. We'll move our way up to some more complex eukaryotic genomes. But one of the areas where we've had a lot of interest and excitement in is sequencing microbial isolates. So if these are cultured organisms, then whole genome sequencing can be performed. But the reason that's quite useful on the Nanopore technology platform is it can be very low cost, it can be accessible and flexible, but also can be assembling complete bacterial genomes. So here you see a beautiful example of a closed circular contig, and you've got a plasmid in there. And again, with short-read technologies, this is very difficult to achieve. And so if you do want to get that resolution, say like antimicrobial resistance genes might be harboring on a plasmid, this gives you that level of resolution and insight, which can be critical for applications like food safety or environmental monitoring or hospital-acquired outbreaks. On the accuracy update side of things, we've made a lot of effort talking to you in the community to understand if there were any errors, and it turns out many of those were associated just with methylation. So we retrained our base calling models to recognize methylation motifs in microbial organisms. And today, that's why we see over Q60 genome accuracy scores. You see an example there from Ryan Wick, who many of you may know is involved in a lot of these assembly tools for microbial genomes. And so we're incredibly excited about that progress. So this can be, again, a standalone platform for bacterial whole genome sequencing, producing not only contiguous, but incredibly highly accurate genome assemblies. So you'll see a QR code on that side that you can scan as well if you'd like to read an application note. And that's a really nice benchmarking data set on foodborne pathogens that we've worked with the FDA CIFSAN group on. Moving on to 16S RNA sequencing. We've seen a few talks throughout PAG talking about microbiome sequencing and 16S. Again, highlighting that when you're able to leverage longer read sequencing, you're able to obtain higher taxonomic resolution. So, of course, historically, sequencing short read sequencing like the V3, V4 hypervariable regions of 16S oftentimes will limit the resolution you can get, maybe at genus level. But if you want higher resolution, and oftentimes species level resolution, then sequencing the longer ribosomal operon is available to you in a highly flexible and low cost and accessible platform. So we do have a kit for this. You can see that this sequences about 1,500 base pairs of 16S, all in a simplified kit that's very quick. And on the accuracy update side of things, we see some great examples from the community, publishing, showcasing the improvements to accuracy, and hitting over Q26, Q28 raw read accuracies, which again are highly amenable for calling these kind of microbiome, even in complex sample applications. Now, as we go even up to the next step with metagenome assembled genomes, I would like to pause here and invite my colleague, Sean Mckenzie, up to the stage, who is our Associate Director of Application Bioinformatics, who's going to talk through a few of the latest and greatest assembly methods. Over to you, Sean.
Thank you, Aaron. Hello, everyone. Today I am excited to share with you how some improvements to community tools for doing de novo assembly, particularly in metagenomic as well as large genome contexts, are giving some of the best results for genome assemblies we've seen yet. So there are two different tools I'd like to talk about, or like to focus talking about. First is for metagenomic genomes. So this is the MetaMDBG2 tool, which was released earlier this year, and a ONT mode was released over the summer. So this is a tool for recovering metagenome assembled genomes, or MAGs, from complex metagenomic samples that you've sequenced with Oxford Nanopore Technologies. And we put this through the test with a true complex metagenome sample, the Zymobiomics fecal reference sample, which is an actual fecal material sample that they've cultured up to a very large batch and aliquoted for long-term reproducibility for benchmarking studies like this. So we sequenced this sample on our PromethION platform, and we found that we were able to generate over 200 gigabases, almost 200 gigabases of sequencing data per PromethION flow cell for this sample. So we generally are seeing that metagenomic samples sequence extremely well with very high throughput on our platform. So we base called this data with our latest Dorado soup models, which, as Aaron just talked about, are now giving us well over Q20 accuracy. And then we put these reads into the MetaMDBG assembler, followed by binning with the new SemiBin 2 tool, as well as a few others, and DOS tool bin consensus calculation. So when we do this, we're able to see incredible results in terms of recovered MAGs. So we got over 298 high-quality MAGs from this sample. And we also obtained at least partial genomes for up to 578 different species, including some of these recovered as multiple strains. So we have at least 770 unique strains in the sample where we're able to recover some form of genome. And again, at least half of these are high-quality mags. When we look at how this performs relative to our other sequencing platforms, we see a clear advantage for long-read sequencing over short-read platforms. With Illumina sequencing, we're not able to assemble a single contig over 1 megabase. And when we look at the number of HQ and MQ mags per gigabase, we see that the performance of these short-read platforms is not competitive. When we compare to other long-read platforms, we see roughly equal performance on a per gigabase level. However, because our PromethION flow cells are not hindered by the shorter read length that you get from microbial communities, because you have to beat to get the gram negatives, we're getting over 200 or almost 200 gigabases per flow cell, whereas our competitors are getting much less than 100 gigabases per flow cell. So on a per flow cell basis, we're seeing many more mags, almost 2 times as many high-quality mags. And this means that your cost per mag is significantly lower for Nanopore Technologies than for any other technology out there. We have a new application note for that, and with a lot more figures and details around that. There's a QR code on your screen if you're interested. Now, moving up in scale from microbial communities to larger, we duplicated our slides for that. So anyway, moving up from microbial communities to larger genome assemblies, there's a very exciting advance last month by Hengli and Haiyu Cheng, which is the porting of Nanopore native simplex support to their Hi-Fi ASM assembler. So previously, earlier this year, we released the Hero and the Dorado Correct pre-assembly correction tool, where you could do pre-assembly correction on our reads, and then these would be appropriate for assembly with Hi-Fi ASM and Virco. So with this new --O&T mode in Hi-Fi ASM, we can now assemble the raw simplex reads without needing any sort of pre-correction, and it turns out to be significantly more computationally efficient than Hero. So these assemblies run in matters of hours on a sufficiently large computer. So we tested this with a wide range of samples, and I'll just showcase 2 today. So one is the tomato, this is a highly inbred variety of Heinz tomato. We threw half of a PromethION flow cell of data at this, generating a 56x coverage data set with a read-in 50 of 30K BP, and we were able to recover half of the chromosomes as telomere-to-telomere contigs, gapless telomere-to-telomere chromosomes, without any form of extra like Hi-C or Poor-C data, and without needing ultra-long. This is just with our simplex LSK data. Now, for larger assemblies, we also see very good performance. So we looked at the Zebu as a large diploid example, and we were able to assemble a highly contiguous genome assembly for this specimen in contig in 50s of 82 and 90 megabases per haplotype. And again, this was a single PromethION flow cell of our LSK reads. We do see very good results with ultra-long reads as well, but I don't have time to show you those except for looking at ultra-long usage for true telomere-to-telomere assemblies in large diploid organisms. So we know for large genomed organisms, ultra-long reads are absolutely essential for telomere-to-telomere assembly, but the traditional way of telomere-to-telomere genome assembly for these large genomes has been a hybrid approach, where you use something like Hi-Fi or duplex reads to build your initial assembly, and then you use ultra-long to help refine that assembly, untangle your assembly graph, and do some scaffolding. But we're now seeing that this is unnecessary to do that initial step. You can generate these genomes just with the ultra-long reads. So we are demonstrating this here with the HG002 human sample, where we generated a 50x ultra-long data set given 2 PromethION flow cells. Base called this with our latest Dorado soup models. And here we're showing pre-correction with the Dorado Correct software, which runs the Hero algorithm, but we're also seeing very good results running Hi-Fi ASM as a pre-assembly corrector and then feeding these results into the Virco pipeline. So we took the pre-corrected reads, fed them into the Virco pipeline along with one flow cell of our Poor-C data, which is the nanopore equivalent to Hi-C. There are some cool advantages of Poor-C over Hi-C, but I don't have time to talk about those right now. Find me if you're interested. But in this context, it functions in basically the same way. It is used for phasing the assembly graph and separating out your haplotypes. Now, we were hoping that with this pre-assembly correction, we would be able to achieve comparable results with just ultralongs as the previous hybrid approaches, and we found instead we were getting much better results. It turns out that building those initial assembly graphs with the ultralong reads is giving us significantly improved results over the old hybrid approaches. So we got over 30 of the 46 human chromosomes as telomere to telomere gapless contigs in the automated assembly, which, as far as we can tell, is the most contiguous automated human assembly out there. Obviously, there's now the Foley T2T HG002 sample, but that required extensive manual intervention. So for automated assemblies, this is the most contiguous assembly ever, and it was actually produced with less data than other T2T assemblies. And we believe that this is generally applicable to any large diploid organism. All right, and so now I will turn it back over to my colleague to present on some of the platforms that can enable you to generate this data to produce these highly contiguous and T2T genome assemblies.
Thank you, Sean. Can we have a round of applause for Sean, please? Excellent. So very briefly, as Sean just highlighted, there are some amazing advancements to the technology, either the underlying accuracy or the assembly tool. So please do check it out and come speak with us if you have any questions. But as you can see, there is also a diverse range of platforms that are suitable for different needs, whether you're looking for something that might be more personal or deployable, maybe the Mark-1D, or a benchtop instrument like the GridION, all the way up to our P2 profiles. So these can host 2 of our Promethean 2 flow cells, either run one at a time or run both at a time, all the way up to our flexible P24 system. And then for those of you who might be interested in automation needs, we also have the Elysian platform. So this can go from sample all the way to answer. And what I mean by that is even putting in the sample itself, having all the extraction on board, and even loading and washing the flow cell itself without any manual intervention. So it's even got a little robotic arm where it'll like open up the port and never introduces air bubbles. So without further ado, this is just more of a concluding slide as we launch into our amazing speaker presentations. But we hope we've given you a little bit on the update side of things from Oxford Nanopore Technologies, working really hard to be a standalone and solid platform for all of your biological needs. Again, whether that's all the way from SNPs to structural variants or epigenetic information, all the way to T2T as a comprehensive standalone platform. So thanks very much for listening to some of those updates.